All right, let's dive right into this explainer and explore the fascinating yet honestly incredibly delicate human respiratory system. I mean, it's a system so sensitive that it can be thrown completely off balance by something as bizarre as hot tub lung, which, yes, is a real condition caused by mold growing in poorly maintained hot tubs. Our lungs are absolutely essential for survival, yet their membranes are astonishingly fragile. So today, we're going to master exactly how this system works, how it's structured, and most importantly, how to conquer it on exam day.
Now, you might look at this number and wonder, why are we dedicating a whole explainer to this? Well, while the respiratory system only makes up about 3% of MCAT biology questions, the crucial point here is that it perfectly integrates high-yield concepts from other subjects, specifically physics and general chemistry. You really can't understand the lungs without understanding work, pressure, gases, and equilibrium. Mastering this system basically means you're mastering applied physics.
Here is our roadmap for today. We'll cover the airflow pathway, the pleura, the mechanics of breathing, lung volumes, and finally, regulation and a quick MCAT review.
Okay, let's jump straight into Section 1, the airflow pathway.
Let's actually trace the structural flow of a single breath. Air enters the nares of the nose, where it gets immediately filtered by mucous membranes and nasal hairs, which we call vibrissae. This warms, humidifies, and cleans the air. From the nasal cavity, it passes into the pharynx, which is a shared pathway for both food and air, and then down into the larynx, which is exclusively an airway. Now, what's super cool about this structural flow is the specialized built-in security system along the way. To keep you from choking, the opening of the larynx, the glottis, is covered by the epiglottis during swallowing. After passing the vocal cords, air travels down the cartilaginous trachea, which then splits into two main stem bronchi. Both of those are lined with ciliated epithelial cells to catch any leftover dust. Finally, the air reaches the bronchioles, ending in tiny balloon-like structures called alveoli. Each alveolus is coated with surfactant, which is a vital detergent that lowers surface tension to prevent these delicate little sacs from collapsing on themselves.
Moving right along to Section 2, the pleura.
Your lungs don't just float freely in your chest cavity. Each lung is surrounded by a closed, double-layered sac called the pleura, and they're bound by this sac against which they expand. You have an inner layer directly hugging the lung, known as the visceral pleura, and an outer layer lining the chest wall called the parietal pleura. Right in between them is the intrapleural space, which contains a thin layer of lubricating fluid.
To really easily visualize this for the exam, picture a fist pushing into a partially deflated balloon. Your fist is the lung. The balloon surface, stretching directly over your skin, that's the visceral pleura. The outer layer of the balloon facing away from you is the parietal pleura, sitting against the chest wall. And that tiny bit of air trapped between the balloon's rubber layers, that perfectly represents the fluid-filled intrapleural space. This closed system is absolutely critical for how we breathe.
So how does this anatomy actually draw air in? That brings us to Section 3, mechanics of breathing.
First off, the lungs do not fill passively. They actually require skeletal muscle to generate pressure. During inhalation, the diaphragm, that thin muscle at the bottom of the chest, contracts and flattens downward. At the very same time, the external intercostal muscles between your ribs contract to pull the chest wall outward. This expansion physically increases your intrathoracic volume. Because the intrapleural space is right up against the chest wall, its volume increases first. And here is where Boyle's law makes a star appearance. According to Boyle's law, at a constant temperature, pressure and volume are inversely related. So as the intrapleural volume goes up, the intrapleural pressure goes down. It literally drops below atmospheric pressure. The lungs, which are sitting at atmospheric pressure, are then sucked into this low-pressure space, expanding their volume and dropping their own internal pressure. Finally, outside air is sucked in to balance it all out. It's a beautiful domino effect.
So the big takeaway for your test here is this comparison. Inhalation is always an active, energy-consuming process driven by what we call negative pressure breathing. Exhalation, on the other hand, is usually totally passive during calm states. You simply relax your diaphragm and intercostal muscles, the chest cavity shrinks, the intrapleural pressure rises, and the intrinsic elastic recoil of the lungs forces the air right back out.
All right, let's hit Section 4, lung volumes.
Let's translate those physical mechanics into the actual data physicians measure on a spirometry graph. Total lung capacity is the absolute maximum volume of air your lungs can hold. Tidal volume is the small standard amount of air you move in and out during a normal, chill breath.
And the maximum air you can force in above a normal breath or force out after a normal exhalation, well, those are known as your inspiratory and expiratory reserve volumes, respectively.
Let's test your critical thinking with a classic exam trap. Think back to the balloon analogy and the mechanics we just went over. Based on how a spirometer works, basically measuring the air you blow in and out of a tube, what essential long metric can it absolutely not measure?
Did you guess it? It's residual volume. A spirometer cannot measure residual volume, which is the baseline amount of air permanently trapped in the lungs even after the most forceful exhalation. This trapped air is vital, guys. Without it, the alveoli would completely collapse and stick together, making it nearly impossible to inflate them again. Because you can never exhale this air into the machine, the spirometer can never directly measure it.
Let's wrap things up with Section 5, Regulation and a quick MCAT review.
Breathing is mostly on autopilot. Your ventilation center, located in a part of the brainstem called the medulla oblongata, runs a continuous feedback loop. Interestingly, it isn't primarily looking at oxygen, it's mostly monitoring carbon dioxide. Chemoreceptors detect high carbon dioxide levels in the blood, a state called hypercapnia. When CO2 spikes, the medulla immediately forces an increase in your respiratory rate so you can blow off that excess gas and restore balance.
Okay, let's tackle an actual passage-style question you might see on test day. If a patient is diagnosed with emphysema, a disease often caused by smoking that destroys the alveolar walls and decreases the elastic recoil of the lungs, what happens to their lung capacities?
The answer? Increased residual volume. Remember how calm exhalation is a passive process relying purely on the elastic recoil of the lungs? Well, because emphysema destroys that elasticity, the lungs essentially become loose and floppy. Passive exhalation becomes incredibly difficult, leaving much more air trapped inside the lungs after every single breath. This inherently increases the patient's residual volume.
Let's try one more clinical application, this time at the very beginning of life. Why do premature infants often require specialized ventilators that maintain positive end expiratory pressure? Like, what is structurally missing?
The answer is that premature infants are born before their bodies have produced sufficient surfactant. Surfactant acts as a biological detergent to lower the surface tension in those tiny alveoli. Without it, the surface tension of the fluid lining the alveoli is so high that the air sacs completely collapse upon exhalation. The ventilator is required to physically force the alveoli to stay open until the infant's biology can catch up.
I really hope this explainer brilliantly illustrated how structural anatomy utilizes simple physics like Boyle's law and surface tension to keep us alive. The respiratory system is just a masterpiece of pressure gradients. Keep these rules of pressure and volume in mind and you will absolutely crush your biology passages. It leaves us with a fascinating question though. If the simple physics of pressure drives our breathing, where else in the complex machinery of the human body does raw physics directly dictate our biology? Think on that, and until next time, keep learning.